Hello, how are you? This is our second plant. Okay, so at the end of the presentation, we should be able to recognize from microscope slides or electron micrographs. We should be able to recognize and draw red blood cells, white blood cells of special mention, the phagocytes, which are the monocytes and neutrophils, and the lymphocytes. We should also be able to determine and state the function of water being the main component of blood plasma and its role in transport in mammals. I'm sure we remember this from our section in biological molecules. And then we also want to find out how tissue fluid is formed and its functions in a capillary network. Now, blood is made up of four major components. We want to be able to determine what each of these four major components do. Firstly, it's made up of plasma, which is the liquid portion, that is blood minus all these cells. Then it's made up of the red blood cells, also called erythrocytes. It's made up of white blood cells, also called the leukocytes. And it's also made up of platelets, also called thrombocytes. The red blood cells, kind of, look at them, they look kind of donut shaped, okay? This shape enhances the surface area because this infolding also enables the cell to have a larger surface area and also for withstanding pressure and for easy flow in capillaries. Again, you can see that from your knowledge you should be able to know that a mature red blood cell has no nucleus, so as to create additional space for hemoglobin, which means that the loss of the nucleus will also mean that the cell has got a short lifespan, and this cell can only last for about 120 days, after which it is destroyed in the liver and spleen and recycled. The iron is taken up to build new cells, and the other fragments are used to form bile. Okay. The hemoglobin inside the red blood cells is used to reversibly bind to oxygen, forming oxyhemoglobin, and thereby transporting oxygen around the body. So the oxygen is picked up in the lungs, transported to the body, and then they pick up carbon dioxide, the red blood cells, and bring it to the lungs where it is exchanged. So it implies that because these red blood cells have got a short lifespan, they are continually being formed in the red bone marrow and being destroyed after they've reached their expiration date. Okay. Of the cellular content in the blood, these make up the bulk of the cellular content in the blood. Of special mention, blood is red due to the presence of hemoglobin. So hemoglobin, you know from our lessons in biological molecules, hemoglobin is a complex protein made up of 4 protein strands, 2 alpha chains and 2 beta chains. And these polypeptide chains are bonded to heme groups, which contain iron. And each hemoglobin molecule can carry up to 4 oxygen atoms. And the presence of this ion in the hemoglobin makes the presence of oxygen in the hemoglobin makes the hemoglobin bright red, and thereby giving oxygenated blood its characteristic red color. Okay. So we say that the other red blood cells live up to about 120 days, after which they are recycled in the liver and the spleen. So the production of red blood cells is facilitated by this hormone, erythropoietin, and this hormone is made by the kidneys, and it works in the red bone marrow. Okay. okay, whereby it facilitates the production, or stimulates rather the production of red blood cells. All right. So, as a something to think about, if your diet is poor in iron, what do you think will happen to your red blood cells? Do you think you make fewer red blood cells because there's less iron to make hemoglobin, or you make more red blood cells to make up for the lack of iron in the hemoglobin, or you just nothing will change? Okay, you've got what do you think? Obviously, you make fewer red blood cells, isn't it? Because there's less iron to make hemoglobin, which is needed in the red blood cells. So, it is interesting that some of the athletes at the Olympics use erythropoietin as a hormone to boost their performance. What do you think this hormone helps them to do? Food for thought. The other group of cells are the white blood cells. There are two types. There are the phagocytes, which are the ones which ingest by endocytosis or phagocytosis and destroy bacteria. And then there are the lymphocytes, which produce antibodies to destroy, which then therefore destroy the pathogens. And blood cells come in different shapes and types. Of special mention to our syllabus are the neutrophils. You can see that the nucleus is a bit lobed, and then we've got the phagocytes. We also have these monocytes. They've got a very large nucleus. You can see that the difference between a neutrophil and a monocyte is that this one seems to have a lobed nucleus, the neutrophil, and the monocyte seems to have a very large nucleus. But the lymphocyte is a bit smaller than the phagocytes, and its nucleus, although it is large, it is kind of sort of bean shaped here. Okay, take note of those differences. And then the platelets are derived from a group of cells called megakaryocytes, and these ones they do not have a nucleus, and that therefore implies that they've got a very short lifespan, which is usually about 10 days. Okay, so these platelets are used in blood clotting. They aggregate at a wound, and then they produce some chemicals which stimulate a production of some clotting factors or clotting proteins, and then they help in clotting. The main one is the red blood cell. So if a person had a defect in the gene for fibrinogen, the obvious thing that would happen is that the person would have problems in blood clotting, isn't it? Okay. And then the plasma is the liquid portion of the blood, which is like a solution or suspension transporting water with the dissolved mainly dissolved substances, e.g. glucose and amino acids, etc. Also, due to the high specific heat capacity, it means that this water enables the transport to be facilitated well, because the water will not change temperature rapidly, and hence the substances, or at least substances being transported, can be transported at a constant temperature. All right. And then the nutrients, we've got plasma transport nutrients, e.g. glucose, salts, etc. And then of course there are the plasma proteins, which like fibrinogen and other blood clotting proteins, some albumins as well, etc. And then of course transport of hormones, e.g. insulin, controlling body functions, and then of course some waste like urea, which is excreted. So tissue fluid now, Tissue fluid now is a fluid surrounding cells of a tissue. It supplies these tissues or these cells with all their oxygen and nutrient needs, and removing all the waste, especially carbon dioxide. So tissue fluid is also important in substance exchange between the blood and the cells. And plasma from the blood capillaries move to the tissue through gaps in these walls of the capillaries, and they become tissue fluid, and they exchange their oxygen and nutrients with the cells, and then they take up the waste. This is of course an oversimplified diagram, these ones representing walls of the capillaries, and within these walls are gaps through which nutrients can escape, but the blood cells cannot escape because the walls, the gaps in the walls are too narrow, so the cells can continue moving. And you can see that these walls are narrow, so they can only allow one cell at a time, and hence this kind of slows things down, and hence enables maximum absorption or maximum exchange of substances. So at the end of the capillary bed, the tissue fluid leaks back into the blood and then becomes plasma again, although not all of it is going to be absorbed. Some of it is absorbed by the lymphatic vessels and becomes lymph. So this is a more correct diagram representing what happens in the tissue fluid. We've got arterial bringing blood to the capillary bed. We've got the arterial dividing into a network of capillaries. Then the blood plasma leaks into this area, okay, and it becomes tissue fluid. Then the tissue fluid exchanges its contents and continues to move, takes up waste. Then at the end of the capillary bed, the tissue fluid is full of carbon dioxide and waste and leaks back again through some gaps, and then it becomes a vein now, a venue, which will join a vein and then bring back the waste to the lungs and wherever they are needed for excretion. So in summary, we were able to look at red blood cells, white blood cells, and from photomicrographs and diagrams, and you were able to at least label them. And we also saw that water is the main component of blood, especially blood plasma, and its properties enable it to be used as a solvent and as a mediator to facilitate homeostasis. And then of course we saw that as the substances are being exchanged in the capillaries, tissue fluid is formed and helps exchange, and then the capillary network is also formed, which further enhances the exchange of materials. Okay, so thank you for listening. I hope you benefited much. Let's meet in the next video, which will be posted. If you have got any questions, please feel free to ask.